Sorry for the confusion about my participation here, but thanks for having me on. I'm Gaetan. I'm going to be talking about my work, CpG Transformer for the imputation of single-cell methylomes. So let's set the stage. Single-cell bisulfite sequencing is being used to profile DNA methylation in single cells. And yeah, I think many of you will know that DNA methylation, it's an important biological process. It's been involved in gene regulation, developmental processes, and also cancergenesis. So of course, it's interesting to profile that in single cells. So that's what I have depicted here on the left of the slide. So that is a bisulfite sequencing experiment. So reads aligned to the genome, and if they are treated with bisulfite, you can profile or you can know that a read has on a CpG site is methylated or not. So that's in blue and red. So either CpG site in a cell can be unmethylated or methylated. So on the computer, we represent that as a partially unobserved matrix. And in this example here, the unobservedness really is quite okay. So there are not that many question marks, but in reality, if you would do this, it would be way worse. And that, of course, is a problem. So that is what CpG Transformer is designed to tackle. It will try to impute those question marks. And okay, so before we go into the model, let's talk about two biological assumptions or rules that we know from just biological knowledge that we can use in our model. So this is on the bottom of the slide. So first of all, we know that CpG sites that are close to each other on the genome are somewhat correlated somehow. So more often than not, they will have the same label or methylation status. And then also, normally, if we do an experiment, cells will be correlated as well, because most of the time we profile cells that have the same function or come from the same differentiation stock. So we can also model correlations between cells. So that's what we want to model. How do we formalize this? First, let's talk about the inputs to CpG Transformer. So we operate directly on the methylation matrix. So a 2D input, much like an image would be a 2D input. And we create a vector for every matrix entry. So just like an image would have also a vector for every pixel, which contains the RGB values, so a three-dimensional vector. We also want here, for our matrix, we also want to have a vector. So what kind of information should we encode into this vector? So first of all, we have the CpG embedding, which just encodes whether that matrix entry was methylated, unmethylated, or unknown. Then also we can communicate the location row-wise with a cell index embedding. So this is applied row-wise, so every matrix entry of the same cell has the same cell index embedding. And because we add this to our model, we can kind of learn a clustering between cells, kind of an implicit clustering. And then we can also add column information of the CpG sites by taking the DNA context around the CpG site, putting that through a CNN and getting a vector, a DNA context embedding. So to briefly recapitulate, we have a vector for every matrix entry that contains three things. Basically, what was the label in the matrix entry, then what was the location in the row, and then also some kind of location column-wise encoding as well via the DNA context. So that brings us to how do we then use these inputs to model, basically these biological assumptions that I just mentioned. So for that reason, we use self-attention-based information flow. So it's a transformer. And I might hear you thinking that a transformer, right, there for 1D inputs for sequences, language, or proteins, or DNA, whatever. How do you use this for a 2D input like we have here? And basically, we solve that issue by iteratively applying self-attention across rows and then also across columns. So this is a concept called axial attention. It's I think introduced somewhat 3 years ago, and it's also incidentally the same kind of technique that has been used in AlphaFold. So in AlphaFold, we do not operate on a methylation matrix, of course, but on an input MSA. So the columns would be amino acids, and the rows would be the proteins in the multiple sequence alignment. So it's basically the same trick that we're using here. Also, for for the first, so to model interactions between the different columns per row, we use a sliding window self-attention approach. And then also, why do we use transformers? I'm going to have to skip the explanation for that because in the interest of time, but you can always ask me later. So yeah, that's the model. So how do we train this whole thing? For that purpose, we take some inspiration from the field of natural language processing, more specifically the bird language model, in which we have a sentence, then we mask some words in that sentence, and then we ask the model, okay, predict what these words were. And I think there's an interesting parallel with imputation. So here we have a DNA methylation matrix. We randomly, at every training iteration, we randomly mask some observed elements. And then we ask the model, okay, what are those elements supposed to be? And so this is basically just a classification task. And then at inference time, after we have trained our model, because our model is optimized to predict from the question marks the labels, we just skip the masking step. We give to our input just our methylation matrix, and we get an imputed matrix back. Right, so results, I think I can also be quite brief here by just saying, I think we all know that I wouldn't be standing here if the model was complete rubbish. So I tested the CpG Transformer on 5 data sets and also compared them with 2 competing models. That also, yeah, it's not unimportant to know, these 2 competing models, they kind of use the same biological assumptions. So they also model correlations between rows and also model correlations between columns in the same row. But yeah, bold is good. So my model scores the best on all of these data sets. And it's also not unimportant to mention that CpG Transformer also outperforms other models in biologically relevant contexts, such as CpG islands and transcription factor binding sites and promoters and whatnot. So these are 2 other nice to haves, which I will briefly mention. These are described in our paper and also implemented at our GitHub repo. So if you are a biologist wanting to impute your own methylation data set, it's nice to know that we can apply transfer learning to this whole thing. And what do I mean with that? Is that we publish the trained weights of the models on these 5 data sets. And if you then want to impute your own data set, you can transfer the weights initialized on these other data sets to impute your own data set, and that drastically reduces the training time. And also the model is quite interpretable. So using these fancy explainable AI tools, we can ask the model, why did you predict a certain thing? So in this example on the right, there's a question mark. It predicted that it should be a 1, and then we can ask why. And then in this example, it says, okay, there are some neighboring CpG sites which also had a 1, which kind of makes sense, of course. So that's all. You can find our paper on bioinformatics, thanks to my co-authors, thanks to my funding agency.